Okay, perhaps we should start. So welcome everyone to attend today's Monash Biomedical Imaging webinar. I would like to start our webinar by acknowledging the people from the Kulin Nations on whose land Monash University is positioned. I pay my respects to their elders, past and present. So before we start our presentation, before we start introducing our speakers, just a housekeeping item. Please ask your questions for the speakers via the Q&A button. So these questions will be answered at the end of the speaker's presentation. The chat section has been disabled, so questions must be sent through the Q&A. Now, it's my great pleasure to introduce our speakers today. So we have two excellent speakers today. Our first speaker is Dr. Brett Paterson. Brett is a National Imaging Facility Fellow at Monash Biomedical Imaging and the academic group leader in the School of Chemistry at Monash University. So he leads the chemistry, he also leads the chemistry research lab at MBI. So Dr. Paterson was awarded a Victorian Fellowship in 2011, which he used to conduct research and training in radiochemistry, biology, and the molecular imaging at the King's College London and the Boston Children's Hospital and Harvard Medical School. So his current research focuses on the development of radiochemistry and radiopharmaceuticals. Welcome, Brett. Our second speaker is Cormac Kelderman. Cormac is a PhD student at this moment under the supervision of Brett in the School of Chemistry and the Monash Biomedical Imaging as well. So his research focuses on developing new chemistry and radiochemistry for imaging and the therapy. Welcome both. So without further delay, I will hand over to Brett for his presentation first. And then Brett, if you can hand over to pass it to Cormac at the end of your presentation. So thank you both and looking forward. I'll stop sharing and pass on to you.
So let's make sure I'm sharing the right screen here.
Yeah, sure. It's always the issue is multi-display.
Can you see that?
Yeah, I can see it.
Can you see it? Oh, you can see it. Oh, excellent. All right. Sorry about that. So yes, first of all, I'd like to thank Charlene for that introduction. That was very nice of you. And thank you for the invitation to talk today about our work. So first, I'd just like to acknowledge some people. Cormac is going to be talking today. He's been an excellent student of mine, and I'm sure you'll enjoy his part of the talk. And I'll be also talking a little bit about a student of mine, Rachel, who has just finished her honours year and is about to start her PhD. I'd like to thank the rest of my group in the School of Chemistry. I'd like to thank everybody at MBI, especially Gary, Mike, and Katie. I'd like to thank the NIF as well for the fellowship and for supporting our chemistry, and the ARC and the Victorian government as well. So a little bit of an introduction on what we've got at MBI in terms of radiochemistry. So we have a hot cell, as you can see here. Inside the hot cell, we often have two gallium generators. These are ex-hospital generators, and that allows us to access gallium-68 on a daily basis. We also have a license for copper 64 and zirconium 89, which are positron emitters, technetium 99M, which will be the focus of a lot of what you'll hear today. And we also have a license for lutetium 177 as well. On the next photo here, we have a picture of our radio HPLC and the radio TLC, which sits next to it. These analytical tools allow us to identify and purify our compounds. This figure on the right hand side here is just what you get from a HPLC. So we have the retention time on the bottom and counts on the left hand side, and this is essentially showing that 95% of the compound of the mixture is eluting in that major peak, and we have a small amount of an impurity there. Once we've got our compounds pure and analyzed, then we can do some preclinical imaging at MBI. So we have the Invion, which incorporates PET, SPEC, and CT. We get an image. This is actually a video, but it's not working for me, unfortunately. But essentially, it's showing this spot here is uptake in the carotid artery using PET, and then that's overlaid with a CT scan. Then we can also take those animals, we can dissect them, take the organs and count them using the gamma counter, and that can tell us, we can quantify each of the organs, the uptake in each of the organs, and can tell us a lot more about the biodistribution of the compounds, and we get a plot that looks something like that. So the radiation that we're interested in therapy and diagnostics are gamma emitting isotopes. So a gamma ray is emitted from the nucleus, or it may eject a positron, which will then annihilate and form two high energy photons. And then these photons can be detected outside of the body and turned into an image, either using SPECT or PET. And then we're also interested in therapeutic isotopes that may emit alphas, betas, or Auger's. These essentially work the same way. They create reactive oxygen species in the body, which can then damage DNA and can be used to treat cancer. So this is a periodic table. I'm sure you would have seen these before, but highlighted in the gray are the isotopes which have elements of isotopes that are of interest to us. I've just highlighted some of the ones that you may have seen before. So carbon and fluorine are used in PET imaging. But as you can see, as you go down the periodic table into the metals, there's a whole range of different isotopes which have properties that are of use to us. So copper, for instance, has a positron and a beta, so that can be used as a theranostic, same as gallium. And then we have technetium here, and then these ones in blue, so lead, bismuth, radium, and actinium are alpha emitters. And then right down the bottom here that I've highlighted in green is lutetium, which is a beta emitter. Some examples of some radiometal-pharmaceuticals that have been on, that are available. So this is technetium 99M HMPAO, also known as Ceratec. So technetium is incorporated into this PAO chelator, forms a neutral lipophilic complex that can be used to image blood perfusion in the brain. And then, so this is just an image here of the specs and then overlaid with the MRI. In terms of therapy, there's a compound which is only recently been approved by the FDA. It utilizes the Peter-emitting isotope Technetium-177. It's incorporated into a macrocycle, a tetraazo macrocycle called DOTA, which makes it nice and stable, which is important. And then that's been attached onto a targeting peptide, in this case, octreotate, which has high affinity for neuroendocrine tumours. So on the right-hand side here, we have a gallium dotatate scan. So this is a PET scan, and this is a patient with metastatic neuroendocrine cancer. And you can see that they've then had the therapy with lutetium-177, and then a month later, the PET scan shows that there is a marked loss of the lesions. Another compound which I've had a hand in making and developing is copper sartate. So in this case, instead of the tetraazo macrocycle, we've incorporated a sarcophagene, which forms a very stable copper complex. And again, we've attached that to octreotate in this case. So as I was saying before, copper has isotopes, a positron emitting isotope, copper 64, but also has a beta emitting isotope. So the copper 64 PET images, this is an example here of a mouse with a xenograph showing uptake in the tumor after 4 hours. And then we can take those mice, and if we inject them with the copper 67 variant, this is just a plot showing that this slows the tumor growth compared to the control, and is very similar to what you would get with lutetium with the lutetium dotatate. On the left-hand side here, we have some first-in-man images that were taken at the Peter Mac with copper sartate, and it's compared to the gallium dotatate, and they are quite comparable. And now this compound is now entering into phase 2 trials. In terms of alpha therapy, the field has really kind of grown in the last few years with the introduction of radium-223 chloride, one of the most toxic drugs on the market. Essentially, the radium is an alpha emitter and acts a lot like calcium, so it has quite good bone uptake and can be used to treat quite severe pain in patients. And another alpha emitter is actinium-225, just showing the effectiveness of this isotope. So it's been incorporated again into a tetraazo macrocycle. This time, at the targeting moiety targets prostate cancer. And so this is a PET scan in A showing the lesions of this metastatic prostate cancer patient. This patient has had 3 doses of actinium PSMA, and you can see quite a remarkable loss of all those lesions. So what we do in my group, some of the work we do is try to develop new chelators for radiopharmaceuticals. And these are some of the properties that we try to look for. So the main one, I guess, is to make sure that the complex is stable. So when we inject it into the patient or in preclinical trials, we want to make sure that the isotope stays inside of the radiopharmaceutical. So we need to be inert as well as stable. We want them to react quantitatively with radiometal ions, low chelator and radiometal concentrations. So the radiometals come in low concentrations, and we want to make sure that to make the process as simple as possible. If it's quantitative, we don't have to do a purification step. Again, the same thing with the kinetic. So if it's fast, then it just simplifies the process. And if possible, we want to be able to attach on a targeting molecule such as a peptide or an antibody. So a few years back now, as part of my PhD, I synthesized these variants of ATSM. The ATSM shown here is a ligand that coordinates copper and various other metals. And so what we did was attach a linker to the ATSM and then incorporate a peptide. This one is bombesin 7-14, and we made the copper complexes of that as well, both the cold copper and the radioactive copper, and just the HPLC in the right hand side here. So the black trace is the radioactive trace and the red trace is the non-radioactive. So because they're eluting at the same time, we can infer that from that they have the same structure. Fortunately, this compound ticks 3 of the boxes along here, but the copper complex, the complexes are just not kinetically inert enough to be of too much practical application. And so then this moves on to some of the work that we're doing in our group now. So over the years, people have tried to radio label ATSM with technetium, and this is the way they've normally done it. So they just take the technetate, which comes off the generator, add a reducing agent, and they heat it. And there's not really much thought involved in it, and the characterization has always been quite poor. Then a group in Italy about 3 years ago published this work where they took the technetate and they reduced it using tin chloride again. This time they added in succinic dihydrazide, and that was able to form a stable intermediate, and then which was a nitrido, and then they could react that with the ATSM at pH 6 for 85 degrees for half an hour to get a compound that they could characterize quite well, that incorporated the chelator, which is N2S2, and then it has this apical nitrido group as well. So what I asked my guys to do was, so Cormac and Rachel, was to repeat this work, and then they could show that we could get really quite stunning purity with these compounds just by copying what this group did, which was really nice. But they didn't stop there. They thought, this is silly. Why is it 2 steps? Why do we have to purge it with nitrogen? Why does it take so long? So they went away and they optimized it. And they basically could show that just by adding in the SDH, the tin, and the ATSM into one pot and heating it 85 degrees for just 10 minutes, they could get essentially the same radiochemical purity. They could even show, so this plot down the bottom here is concentration versus purity, and they could show they could get the concentration of the ATSM right down to 1 micromolar and still have 100% radiochemical yield. So the optimization, now we're down to a 10-minute reaction time. The concentrations are much lower, and and it's only in one step, so it has the advantages of being incorporated into a kit form, and we didn't need to purify to actually purge it with nitrogen either. So Rachel in the group then went away and made a whole bunch of different variants. I'm not going to show the structures here, but she incorporated different linker groups and incorporated a targeting molecule as well. And she could show that each of these had a HPLC showing excellent radiochemical purity. And then could also show that each of these linkers could change the hydrophilicity of the compound. So copper ATSM up here is quite a hydrophobic compound. The copper complex is used actually to image the brain. So these are quite hydrophobic. This is the plot here of log D. So it's the ratio of uptake in organic solution compared to an aqueous solution. So positive value means it's going into the organic layer, whereas a negative value means it's much more hydrophilic in this case. So that and so this is important because most radiopharmaceuticals that use a targeting molecule are quite hydrophilic. And now the moment of truth was to see how stable these were. And so they did a competition experiment where they added the compounds to either cysteine, a 0.1 millimolar, or into serum, into human serum. And this plot is just got radiochemical purity from 0 to 100. And as you can see over time, for the cysteine, for both of the compounds, they attained a radiochemical purity of close to 100%. And we did see some loss of the purity in human serums, but it's still up to 24 hours, still greater than 90% intact. And this is just the HPLC showing that. So we have the complex here eluting somewhere around 28 minutes. If it was incorporated in the human serum, it would come out a lot earlier. So we're just seeing a small little peak there showing that it's come out. So this time with these technetium compounds, we were able to actually tick that box and say we have something which is thermodynamically and kinetically stable. And so the next step now would be for us to determine if we can produce a signal to noise that would be good for imaging. So that is the next step in this work. So with that, I think my time might be up. So I'm going to hand over to Cormac and stop sharing. Over to you, Cormac.
Yes, I'll just share my screen. So can everyone see that all right? Cool.
Yeah, you can see it.
Okay, so yes, thank you for inviting us to the webinar and presenting. And thanks, Brett, for that great introduction. It really will help people sort of understand what I'm talking about. So my section of this webinar is to present some work I've been doing on technetium 99M bis-thiocemi-carboxylato complexes for SPECT imaging. So just first of all, a layout of the presentation. I'll start off with a brief introduction on both technetium and bis-thiocemi-carboxylanes. Then as a chemist, I'm obligated to discuss a little bit of chemical synthesis. So I'll go through that and then focus a bit more on the radiochemical synthesis in vitro experiments and finally the in vivo experiments. So just starting off with an introduction. as Brett mentioned, Technetium-99m has a number of clinical radiopharmaceuticals, and they utilize single photon emission computed tomography or SPECT imaging. So just here I have a number of different clinical Technetium-99m drugs. So starting off with some kidney drugs, and then Paterson, which is a thyroid drug, the cardiolite, which is a heart imaging drug, and then Ceratec, which is a brain imaging drug, which is one that Brett showed in his slides. So just for technetium in general, all isotopes of technetium are radioactive and therefore must be artificially produced. So for a number of the traditional inorganic characterizations, we use rhenium, just because it's one row down further on the periodic table to technetium and has very similar chemical properties at a number of different oxidation states. And then finally, Technetium-99m, which is the isotope that we're interested in, can be isolated just from a molybdenum-99 parent isotope using a benchtop generator, as pictured here on the right. So the way that these get used is you elute saline just through the generator over the column, and then that gets sucked up into a vial here, whereby you isolate all of the radiation purely as 99m technetium. So technetium is really popular in nuclear imaging, mainly because of its ease of use with the generator, but also its versatile coordination chemistry, which means that it's being used in about 85% of world nuclear imaging scans, totaling about 40 million annually. So as Brett mentioned about some bis thiochemicarbones focusing on ATSM, most of the work has been done using copper, and Brett spoke a little bit about the work that we've been doing with technetium tetradentate bis thiochemicarbones. However, my work is focused, well, this presentation will focus on the work using hexadentate bis thiochemicarbones. But what we were sort of trying to investigate was, can we alter the biodistribution of bis thiochemicarbones by simple modification of the terminal R groups? So these terminal R groups in this paper, they changed from methyl from ATSM to a number of different amines and polyamines to alter the log D value, which is a proxy for the lipophilicity and hydrophilicity, as Brett mentioned. And they were able to determine that the difference in hydrophilicity and lipophilicity corresponded to a difference in uptake of the radiation in mice. So over here on the right, we have the PET imaging scans of some bulb C mice, whereby the more lipophilic complexes showed higher uptake in the liver and gut system, and the more hydrophilic complexes showed higher uptake in the kidneys and urine. So what we wanted to do was determine if we can repeat those same experiments, but using some technetium bis thiochemicarbazone complexes. So for the synthesis of these hexadentate bis thiochemicarbazones that I've shown here, we start off with a ketone molecule that has a proton in this very labile acidic position, which we can brominate using just bromine to get this bromoketone, which can then undergo an alkylation reaction with a diamine to form new carbon-nitrogen bonds to get a diketone, which is the same backbone, or a very similar backbone to that used in Ceratec, one of the clinically used drugs. So then, instead of following the same synthesis that one would use for Ceratec, we added in two thiochemicarbazide molecules to get this bis thiochemicarbazone. And I've managed to make a number of different analogs of this compound with different, so an unsubstituted R group, so just a proton, and then a number of different alkyl groups and two ether groups as well. So in some previous research, Brett was able to show in some research that he published that with rhenium 5+, these hexadentate bisthiochemicarbozones formed distorted trigonal prismatic structures, whereby you have 4 nitrogen and 2 sulfur donor atoms. So in this research, we wanted to focus on, can that be replicated with technetium, as one would expect from their very similar coordination chemistry properties. So just a little bit on the characterization. I'll try and be as brief as possible because I know that most of this audience is not chemistry background. So just this final step of the synthesis where we go from a diketone to the bisthiochemicarbazone. So this is just the propyl derivative, one of the more simple ones that I managed to synthesize. So here I have a proton NMR, whereby each of these colored dots that I've represented on the molecule can be corresponded to different peaks in the NMR spectra, whereby we're able to determine both that the chemical identity of this product is what we expected, and also that the purity is quite high with very little other peaks in the spectra, with those small peaks that can be seen that don't have dots being from residual solvent from the chemical reactions. And then to further validate that, we do mass spectrometry, whereby we get the mass to charge ratio of the complex and determined that that was indeed the product that we were looking for. So moving on to some radiochemistry. Initially, I started off with the ethyl derivative of my bisthiochemicarbazones, whereby this is the same chelator that was used in the previous literature that elucidated the structure of the rhenium compound. So I took the chelator, the rhenium compound, and some of the initial reaction mixture from one of the first times I did the technetium experiment, and analyzed them all by HPLC. So across the bottom, I have retention time, and plotted against either radiation intensity for the technetium trace in red, or the UV absorbance at 280 nanometers for the green chelator trace and the blue rhenium trace. So what we can see here is that the chelator itself elutes at 6.5 minutes, and then the rhenium complex elutes at 9.5 minutes. So then for what we're looking for in the technetium trace is to have a peak co-elute with the rhenium peak. So therefore we can infer that the coordination environment is indeed the same. And that's exactly what we see with the technetium compound eluting at 9.5 minutes as well. The other 2 peaks in the technetium spectra are pertechnetate, which is just the technetium starting material that we use, and then this secondary intermediate compound, which we suspected and hypothesized was an intermediate based on just some of the properties we had seen in the radiolabeling conditions. So then to determine the identity of this intermediate, we sent some samples to one of our collaborators, Dr. Michelle Ma at King's College London, whereby she was able to radiolabel the chelator with a mixture of technetium-99M and its ground state technetium-99, which allows us to do sort of a bit more traditional chemical characterization using mass spectrometry. So first of all, we extended the HPLC run to get clearer separation between the 2 peaks, blue being the product peak and red being the intermediate, and then analyze the mass spectrum from each of those peaks to determine the identity of the chemical species. So we were able to identify that the mass to charge ratio for the product was 539, as we expected. And then the intermediate was 557.2, which is an additional 18 mass units higher, indicative of an oxygen and 2 hydrogens. So given what we know about technetium coordination chemistry, we expected that that was not just simply a water and was in fact incorporated into the complex somehow. So we did some DFT experiments, which were conducted by another member of the group, Patrick Davey. So DFT is density functional theory, which is a computational method whereby you can determine the lowest energy conformation of a particular structure. So what we were able to do was take the structure that had been identified for rhenium and swap out the metal to technetium and then simulate what the structure would be. So we were able to identify that the technetium structure was almost identical to that of rhenium for the product. And then taking that technetium structure, we then added in the oxygen and 2 protons and found that it gave us a structure very similar to that Ceratec, actually, whereby we have 4 nitrogen donor atoms and this oxygen in an apical position above the plane. So the geometry ends up being square pyramidal, which is very typical for technetium 5+ chemistry. So that was quite interesting and gave us a possible structure that we could present as well. So the next step was to optimize the radiochemical yield by testing how much of the radiation was in the product peak versus total radiation against chelator concentration, temperature, and eventually time as well. So here I have the radiochemical yield optimization of chelator concentration and temperature. So as expected, as you increase the amount of chelator present, you get a higher incorporation of the radiation because there's just more chelator to react. And at higher temperatures, the system has higher energy, allowing for a faster and more efficient reaction to occur. Also at the lower temperatures, we saw more of the intermediate product still being present after the 10 minutes of incubation, whereas at higher temperatures, we saw almost no formation of that intermediate. So one of the sort of ideal conditions we found was 80 degrees, 0.1 millimolar for 10 minutes of incubation. And then we also wanted to see if we could test at 40 degrees and determine a system where we could get higher radiochemical yields to better suit some sensitive biomolecules, such as antibodies and proteins that tend to degrade at temperatures above 40 degrees. So what we were able to determine was that at 0.1 millimolar chelator concentration at 40 degrees, over the course of an hour, we see almost complete reduction of the pectinitate starting material and this intermediate peak at just after 10 minutes to almost exclusively the product after that 60-minute period. So then taking those 2 conditions, so the bottom trace and then the second trace here as well, we were able to determine some optimum conditions. One final thing we did was determine if we could achieve really high purity with a lower chelator concentration at 80 degrees, whereby we see no formation of the intermediate, and then purify it using a solid phase exchange cartridge. And we were able to determine that at 80 degrees for 10 minutes at 0.01 millimolar chelator concentration, we could achieve almost 100% radiochemical purity. And each of these peaks also coalesce with the rhenium structure as expected. So taking these conditions, we were able to radiolabel not only just the ethyl derivative, but also all of the others. So the unsubstituted proton, all of the alkyl derivatives, and the 2 ether derivatives as well, giving us retention times between approximately 7 and 13 minutes. So this retention time is based on the relative lipophilicities of the complexes, and that's how they get separated. So one of the things we wanted to test was the different lipophilicities of each of these complexes. So taking each of those, adding them to a mixture of octanol and PBS, we were able to determine the ratio between the ratio of the radiation present in each of those layers, and plot that against retention time. So similar to what I showed earlier for the copper ATSM chelators, we were able to plot these as well. And we found, interestingly, an almost linear trend between retention time and lipophilicity, as shown by the log D value. So we thought it was best to take just 3 of these complexes and analyze them further. So we took the proton, ethyl, and butyl derivatives, as they give quite a nice spread of the retention time and lipophilicity present. So then the next step was to do a cysteine challenge. So similar to what Brett spoke about, we with the Technetium ATSM derivatives, so in this case I used the hexadentates, we were able to show that upon incubation at 37 degrees, in a physiological concentration of cysteine, so just the concentration of cysteine present in human serum, we were able to determine that these complexes were unfortunately quite labile in this system, whereby over 80 minutes we saw almost complete degradation of the compounds, with the radiochemical purity reaching well below 25%. However, looking at these final 80-minute time points, we see a small amount of the final of the product still present. So yeah, between about 10 and 20% left. But interestingly, we see a number of other peaks in the middle, which are all different for each of the compounds. And then this final peak at 5 minutes, which was curious to us because we see it in each of the 3 different spectra, indicating that there's no difference between this compound. So we're able to identify that this was in fact just a technetium cysteine complex by reaction between technetium, between pertechnetate and cysteine. So moving on with this, we then decided to do some mouse planar imaging. So on the left here, we have, so on the bottom, sorry, we have the static 30-minute time point, and then up the top we have the a GIF of the scan from 0 to 30 minutes with slices at 30 seconds each. So each of these images is a mouse with the head pointed downwards and the tail up the top. And then the blue and purple is areas of low radiation uptake, and the red and yellow is high radiation uptake. So for the pertechnetate control, we see almost all of the radiation localizing in the urine, the stomach, and the thyroid, as expected from previous clinical values. So then moving on to the most hydrophilic of my compounds, the unsubstituted proton derivative, we find that most of the radiation ends up in the urine after 30 minutes, but it does swirl around the liver and gut system, and you do see a small amount of uptake in the thyroid as well, which is indicative of breakdown of the complex and it reverting back to pertechnetate. Moving on to the ethyl derivative, we see significantly less uptake in the urine and a lot more in the liver and gut system, and again, just a small amount in the thyroid. Finally, with the most lipophilic of the complexes, the butyl derivative, we see almost none of it going to the urine, and almost all of it staying in the liver and gut system, with a very small amount in the thyroid at the end. So from this qualitative data, we can determine that not only are the complexes different from one another, but they're also different from pertechnetate as well, which is quite nice to see. So in terms of getting some quantitative data, we dissected each of the mice. So we used a number of mice for this and dissected each mouse at 15 minutes. to determine the amount of radiation in each of the organs and base that by weight to get the injected dose per gram. So in this graph, I have asterisks where we have statistical significance of some interest to us, at least. So for the urine, we have statistically less of the butyl derivative, as was expected from the planar imaging that we have. And then for the Technetium control, we see statistically higher amounts of uptake in the thyroid and stomach, again, as we expected from the planar scans. Interestingly, we have higher uptake of the butyl in the liver, which is indicative of where a more lipophilic complex would go. So not too unexpected, but quite nice to see that our complexes are altering the biodistribution. The opposite goes for kidneys, whereby the more hydrophilic complex, the proton derivative, was able to be observed in a significantly higher uptake. And then one thing we weren't necessarily expecting was quite a high uptake in the small intestine, with a statistically significant higher amount of the butyl derivative as well. So just going on to conclusions, we were able to show that we could coordinate technetium 5+ with a number of these hexadentate bis-thiocemic carbozones, and they all formed lipophilic complexes. We then were able to show that the complexes were labile in the presence of a physiological concentration of cysteine over 80 minutes at 37 degrees. And then finally, that we were able to alter the in vivo biodistribution of these complexes through simple modification of the terminal R groups. So just some acknowledgements. I'd like to thank my supervisor, Dr. Brett Paterson, and then everyone else that helped with this research. So Pat, Michelle, Katie, and Mike, as well as the rest of the Paterson group and all the personnel at Monash Analytical Platform and MBI that keep these places running. And then I'd just like to mention that all this research was conducted at the School of Chemistry and Monash Biomedical Imaging, and then was funded by the Australian Research Council and the Australian government. Thanks for listening.
Thank you very much for Brett and Cormac for the excellent presentations. I guess that's really demonstrate the overview of the capability and also the in-depth of labeling and imaging. So really highlights the expertise you have in your group. In view of time, we have, if anyone has burning questions, we can probably answer 1 or 2 burning questions if people have. I guess one of the things I always want to ask to a chemist, where there's a large literature now to look at deep learning to reduce, or machine learning to reduce the dosage in PET imaging in particular. So that makes sense from instrumentation point of view, from physics point of view. But I always want to ask you your view, Brett, in terms of radiochemistry or pharmacokinetic process in biology, whether that really makes sense to reduce the time as well as the dose in the experiment.
That's a really good point. I know I haven't really thought about it. I guess I've always kind of looked at it as a physics problem to see if you could increase the sensitivity. But yes, I guess in terms of what we can try to hope to achieve is to get a better signal to background, so that we can inject less, I guess, and still have enough for an image. So yeah, it's all about getting a better biodistribution, maybe faster clearance from non-target areas, those kind of things. Yeah.
Yeah. I was just going to add just on that. I think that goes along with sort of what I was trying to convey as well, with the tunability of just simple alterations of these terminal groups allows us to hopefully impact those things like clearance rates from non-target organs and things like that to get better signal to noise. So that's sort of one of the points of doing this very minute adjustment, is to get yeah, those better signal to noise ratios.
Yeah, great. Thank you, Brett and Cormac. There's a question on the Q&A. Gary, I saw you on the raise your hand. Do you have any questions? You want to ask a question?
I'll do the Q&A question first, and then
okay, sure. There's a question from Thomas Hampton. So thanks for the fantastic presentation, Cormac and Brett. Do you take into account the distribution of the PET and the SPEC capabilities in the clinics around Australia when planning the experiments? What do people have available here? That's a general question.
Yeah, I think that's a great question. So at MBI, there we have the biograph, and so that enables us to do some large animal and human PET imaging, and we also have the Invion for micro PET, microspec and CT. There's not a lot of micro specs in the country, so we're lucky to have that available at MBI. So yeah, and a lot of those images now people are doing with lutetium and copper 67, doing the biodistributions using SPECT as well is really useful as well. So so yeah, I hope that answers the question.
Yeah, I've got a question to you, Brett, and to Cormac. It's around the resurgence of research in SPECT traces, I guess you would say. And interested in your views, if you like, on the sort of future trends. You know, SPECT's been around a long time, PET of course as well, but there's been a much bigger research effort in PET, I would think, over the last couple of decades. And you know, with the introduction of these very high sensitivity total body PET scanners, you know, which means you can do very, very low dose PET studies. I'm wondering if you think that's going to sort of bounce back and drive more PET pharmaceutical research, or do you see that in fact, you know, the scanners are not the issue? It's more about the sort of particular chelator that you can use and the, you know, the pathological target that you can identify.
Yeah, look, it's an interesting question. I think it's probably a bit of both, Gary. I think, I mean, you know, PET, yeah, spec went out of favor for a long time there. And I think just the availability of the technetium, when it is available, I guess, at least it's widespread, easily available in hospitals, if it is available at all. And I think a lot of people panicked around about that. But at least in Australia now, we've kind of solidified our supply of tech. So I think it's still quite an exciting area of research. But yeah, in terms of the sensitivity of I mean, all we can do is try to create improved radiopharmaceuticals, and I still think that there is a lot of interest in that area, especially with tech, but and then also potentially in the other isotopes as well. And if Cormac has anything to add to that.
Yeah, I think one other thing to sort of add to that is that obviously here at MBI, we're super privileged to have such amazing facilities and instruments, but and the same goes for most places in Australia. We have a lot of access to some of these really high, like super high resolution PET scanning instruments, but that's not necessarily the case all over the world. And SPECT, I think, still just has that much further reaching aspect of it that allows us to conduct research to make these improved radiopharmaceuticals for a much wider audience, I guess, which I think is just something that sometimes can get forgotten, sadly, in the pursuit of sort of better techniques, which tend to be more expensive as well.
Great, excellent. with that, I think we got time, and let's conclude our seminar today. Let's thank our speakers again for their excellent, insightful presentation. So before everyone leaves, our next webinar is on 29th of July with Professor Jason Matney from University of Queensland, and the presentation is entitled Understanding the Role of Prediction in Sensory Recording. Okay, thanks everyone for attending today's seminar, and see you next time. Bye.